So to kind of put a cap on what you drew yesterday, this happens until we hit the stop code, right? That drawing, that kind of comic strip you did. Sorry, I forgot to take your notes out. That dance of the tRNAs coming in and coming through the ribosome two at a time and dropping off their amino acids and the peptide bonds being made, and the strand moving down 3 bases and again and again and again, it happens until you hit the stop codon. Once you hit the stop codon, the amino acid is set free. The chain of amino acids folds up one way or another. Maybe it's already mostly folded, like you saw in the video. Maybe it has an enzyme that comes along and assists with that folding. It was the whole point, that protein was. But the other parts of the process are all totally reusable. And so disassembles, but all those parts can be used again. They don't last forever. In fact, the mRNA that codes for that protein is usually not just getting translated by one ribosome. It's pretty common, and I kind of crudely drew it here, but it's pretty common to see many ribosomes clamped onto one mRNA strand, making the same protein, right? This is the code for one protein, but if you run 10 ribosomes along that mRNA, you're going to make 10 copies of that protein. And so what you'll see is like all these ribosomes along there, and each of them has that protein at a different stage, right? Like this one started first, and it's got the longer one, this one's a little shorter, and so on. Get the idea? So that's kind of the basic story. That story is so central to life on Earth. We actually sometimes call it the central dogma of biology. Dogma meaning the central, thesis, or in a religion, it's like, what's the stuff that's central to that religion? It's kind of a joking term, but it's true. Like this, again, not to oversell things, but like that code, this wheel, like the stuff that codes for the stuff is pretty universal. Okay, before I get into more of that, protein folding is pretty cool. We're not going to spend a lot of time, but it has levels. So the amino acids that are there in what order? That's called the primary structure of a protein. Then there's kind of typical coils and folds that happen. We call those alpha helices and beta sheets. Those are things you see again and again in lots of different proteins. Then the tertiary structure we call just the way it kind of folds up entirely. And then a lot of proteins are made from more than one gene product, and that's catenary structure. When it takes like 2 pieces to make a protein, that's catenary. Here's another version of that. Right, primary, secondary, tertiary, and then it takes more than one. All of that kind of adds up to something like this. This happens to be hemoglobin. You can see a couple of the things we mentioned. These are the helices we were talking about. These are the sheets. This thing happens to be the perfect shape to grab one atom of a particular metal. Do you know what metal? This is a protein that's basically built to hold a piece of metal, which seems weird. It'll make sense when you think about it. That would be iron, right? Iron is what hemoglobin hangs on to so that it can also hang on to oxygen. It's what allows hemoglobin to be an oxygen carrier, to be a part of your red blood cells, which are basically oxygen trucks for your circulatory system. Without that exact shape, it's not going to be able to do that, right? The shape is totally what makes it work. Everybody good? Okay. Here's the thing. That code right there, that wheel, which takes many forms, it doesn't have to be exactly that. That's just the easiest one I know. That thing works the same for everyone. We've never seen a living thing that doesn't use that code.
Interruption, Blake, Trent, would you come to D10 stop us, please?
AUG codes for methionine, whether you are a night crawler, earthworm, whether you're a bacterium kind of thing, whether you're hate, whether you're a potato, all of it uses the same code. It all works the same. So I know that doesn't sound like a lot right now, but I want you to just bear with me for a second. Doesn't that mean that we could take a length of that code from one critter and put it in another and it would work? It does mean that, which comes with an enormous amount of power that like we as humans have right now that we'll get into next chapter, but I kind of want to tease it right now. Doesn't that mean that like if I wanted to take, I don't know, some crazy gene that some crazy power other critters have, like, I don't know, the gene that, it's actually two genes that make a firefly's butt glow. What if I snip those two genes out of the firefly? In a way that meant that I could recombine those two genes with somebody else's DNA. And I don't want to do take just yet. Maybe someday. Let's go with something a little less scary ethically. It's not just the butt, though. Yeah, the whole, yeah, it would be like a whole glow-in-the-dark organism. What if we did that in a cat? Glow-in-the-dark cat? Shouldn't that work? It does. We can. We did. That's real. I mean, yeah. Here. I knew you wouldn't believe me. Not kidding. They did exactly what I just, well, okay. I'm not 100% sure it was firefly, I can't remember. Could have been some bioluminescent like fish or something. But they spliced the DNA, they made something else glow into a cat. Now the catch is you can't do that into a full-grown cat because you'd only be putting it in one cell. So you got to catch the cat when it's only one cell, which means you got to clone the cat. So they cloned a cat from a cat cell that they had spliced that gene into, and they made glow-in-the-dark cats. Nathan.
If you release a bunch of glow-in-the-dark like rats in New York or something, and have the rats going around like next to
The mind boggles. That's awesome. Yeah, so like that's where we're going. You got to learn this stuff so you understand how it works, and then I can give you the tools to make a glow-in-the-dark cat if you want. Not that you should do that. That poor thing is never going to be able to hide from anybody in its life, but it's something you need to be aware of whether you want to do it or not. And I'm totally going to give you instructions on how to do it. You can do it in your kitchen if you want. Yeah. Like, and it's better than the stuff I'm going to show you is actually kind of older. CRISPR is the whole wave of the future. I'll tell you about that too. Point being, what we're going to find is that very quickly we're going to get into things where you're like, why would we do that? And then beyond that, you're going to get into things you're like, we absolutely should not ever do that. And that's what I'm interested in. I'm interested in where the line is between what we can do and what we should do. And that's fun to talk about sometimes. But you'll at least get a chance to kind of establish where is that line for me. Yeah. Main takeaway of the day, is this is the code to all life on Earth, and we know it. Rest of the period, I want you to finish, or at least be well on your way to finishing the paper shape worksheet I gave you yesterday. Including once you get to the end and you have assembled, like, so it's going to have you build a double-stranded DNA, right? You've done that. Then split the DNA and build mRNA onto one half of it. Then you can get rid of the DNA, but you're going to keep that mRNA with the 6 bases. And then you're going to have your tRNAs grab amino acids, and you're going to bring in the tRNA carrying the amino acids, right? Following the instructions, it tells you how to do it. But once you get to that point, why don't you take a picture of it, upload it to the assignment I posted today. The paper copy of the worksheet is something that should be done, but also the upload of the photo. Yuri,
I just had a question on 22.
hang on.
Do you only want to send in a picture of that, or do you want to only send in a picture like a worksheet?
No, I'll check the worksheet in person. That's the beauty of written things. But yeah, upload the photo just so I know you know how that stuff goes together. Cool? Rest of the time is yours. I will